In the summer of 2008, in a limestone cave in the Altai mountains of southern Siberia, a Russian excavation team led by Anatoly Derevyanko and Michael Shunkov lifted a small piece of bone out of a sediment layer. It was the tip of a little finger, small enough to sit on a thumbnail without covering it. Nobody photographed it. The cave had been producing bone for decades, most of it animal, and a fragment that size is not a discovery. It is paperwork. It went into a bag. The bag went into a box, and eventually a sample of it went to a laboratory in Leipzig, in Germany, where a team was reading ancient genetic material out of scraps that used to be considered worthless. What came back did not match anything on the list. It was not a modern human, it was not a Neanderthal. Every other extinct relative of ours arrived the same way. Somebody finds a skull or a jaw or a hip. The bones get measured, argued over, given a Latin name, and then, if the chemistry is kind, a genome comes along decades later to confirm or embarrass the people who did the measuring bones. First, biology second. This one arrived backwards. We had the biology before we had a body. We could describe the shape of their genome to the base pair, and we could not have told you the shape of their face. For 15 years, we had a population of human beings who existed as data and almost nothing else. They have a name now, Denisovans, after the cave. What they do not have still is a settled place in the family tree. And that argument is louder today than it was 10 years ago, not quieter. We are going to work through 5 pieces of evidence in roughly the order they were read, and each one makes the picture larger and stranger than the one before it. And the last piece is not in a museum drawer. It is inside a large number of people currently alive. Number 5. The finger bone that did not fit. Denisova Cave sits above the Anui River in the Altai, a wide-mouthed limestone chamber with a hole in the roof that lets the light down. It is a comfortable place to shelter, which is the whole problem with it. Things that are comfortable get used repeatedly by everyone for a very long time. And the floor of that cave is a stack of everyone's leavings pressed together over roughly 300,000 years. Later work by Elena Zavala and colleagues, published in Nature in 2021, read DNA straight out of those sediment layers and found the pattern. The cave changed hands. One kind of human occupied it, then another, then the first again, with animals in between. It is a hotel register written in dirt. The 2008 season produced the usual mix: bone, teeth, stone tools, most of it animal. Among it was the fragment. The sample went to Svante Pabo's group at the Max Planck Institute for Evolutionary Anthropology. In 2010, Johannes Krauss and his colleagues published the result in the journal Nature, in a paper with a title that says the quiet part out loud, the complete mitochondrial genome of an unknown hominin from southern Siberia. Mitochondrial DNA is the small, separate loop of genetic material passed down the maternal line. It is not the whole picture, but it is abundant and it survives well. And it is very good at answering one specific question: how long ago did this lineage and that lineage last share a mother? The answer for the finger bone was roughly 1 million years. To put that in position, modern humans and Neanderthals share a maternal ancestor somewhere around 500,000 years ago. This lineage branched off before that and then kept going, and was still walking around in Siberia deep into the last ice age. It was not an ancestor of ours, it was not a Neanderthal, it was a third thing, and the third thing had been sitting in a box. And it was not a single freak result, which matters more than it sounds. The cave had also produced a molar, an unusually large upper tooth, too big and too primitive looking to belong comfortably to a modern human or a Neanderthal, catalogued years earlier and treated as an oddity. When that tooth was sequenced, its mitochondrial genome came back closely related to the finger bones and clearly from a different individual. 2 people, then. Not one anomaly. There is a limit here that the paper itself was careful about, and we should be too. Mitochondrial DNA follows one line only: mother to mother to mother. A single unusual mitochondrial lineage can turn up inside a population that is otherwise ordinary. On its own, one strange mitochondrial genome does not make a new kind of human. Krause's team said as much. What they had was an anomaly with a date on it, and the honest conclusion was that they needed the nuclear genome before anyone said the word species out loud. A finger bone gave us a question. The nucleus of that same finger bone was about to give us a population. Number 4. A genome with nobody in it. Later that same year, in 2010, David Reich and colleagues published the nuclear genome in Nature. This is the paper that turned an oddity into a people. The nuclear result did 2 things. First, it placed them. Denisovans were not a lost branch of us, they were the sister group of the Neanderthals, 2 populations that split from each other after both had split from the line leading to modern humans. Cousins to the Neanderthals, more distant cousins to us. Second, and this is the part that stopped the room, the same paper found Denisovan genetic material sitting inside living people, not in Siberians, in Melanesians, the populations of New Guinea, and the islands north and east of Australia, some 9,000 kilometers from the cave. Between 4 and 6% of their genomes traced back to this group. 2 years later, in 2012, Matthias Meyer and colleagues published a high-coverage genome in the journal Science, using a library preparation method that reads single strands of DNA rather than pairs, which roughly doubles the amount recoverable from badly degraded material. The result was a genome sequenced about 30 times over. That is a level of detail comparable to a sequencing project on a living volunteer. From a fingertip, we knew this individual was female. We knew her eye and hair and skin pigmentation were likely dark. We knew which segments of her genome differed from ours and roughly what those segments do. We could even see in the low level of variation carried between her 2 copies of each chromosome the signature the authors read as a small population. A people who were never numerous. We had no idea what she looked like. That gap is the whole reason this episode exists. Everything about the Denisovan body, height, build, the shape of the skull, whether they were heavy-set like Neanderthals or something else entirely, was inference stacked on inference because there was no skull, no pelvis, no long bone. There was a fingertip and, soon after, 2 very large molars. And there is a further crack in the floor here which has widened since. In 2019, Guy Jacobs and colleagues published an analysis in the journal Cell of Denisovan Ancestry in Papuan Genomes. Papuans carry roughly 5% Denisovan ancestry, but not from one source. The Denisovan-derived stretches in those genomes come from at least 2 deeply divergent lineages, separated from each other by more than 350,000 years. That is a gap comparable to the one between us and the Neanderthals, which means the word Denisovan may not describe a people at all. It may be a filing cabinet holding several distinct populations we cannot yet tell apart, because we have never seen most of their bones. So far, every piece of this has come from reading chemistry. The next piece came from reading chemistry too, but what it recorded was not a population or a lineage, it was a household. Number 3. The girl with 2 species in her. In 2012, excavators at Denisova Cave were working through what is politely called unidentifiable bone. Thousands of splinters. None of them diagnostic. The kind of material that historically got cataloged by weight and then left alone. A method changed that. Collagen fingerprinting, known as ZooMS, reads the collagen proteins in a bone chip and identifies which broad group of animal it came from. It is cheap, it is fast, and it turns a bucket of splinters into a searchable index. A team led by Samantha Brown ran it across the Denisova material, and in 2016 flagged 1 sliver about 2 centimeters long as human. The specimen is catalogued as Denisova 11. The people who worked on her call her Denny. In 2018, Viviane Slon, Svante Pabo, and colleagues published her genome in Nature. She was female. She died at least 13 years old, around 90,000 years ago, and her genome was not a blend that had been stirring for generations. It was split cleanly down the middle, chromosome by chromosome. 1 set from a Neanderthal mother, 1 set from a Denisovan father. First generation, not a distant echo. The same paper found something else in her, 1 layer further down. Her Denisovan father was not purely Denisovan either. His own genome carried a trace of Neanderthal ancestry from hundreds of generations before he was born. And her Neanderthal mother was more closely related to Neanderthals living thousands of kilometers to the west than to the Neanderthals who had occupied that very cave earlier, which means populations had been moving across the whole span of Eurasia and meeting at the edges, over and over, long before the night that produced her. She is not the exception in that family. She is the clearest surviving page of it. Consider the odds involved. We have, at the time of that publication, a small number of high-quality archaic genomes in the world, a couple of dozen individuals, total, across 2 extinct populations that lived and died across the whole of Eurasia for hundreds of thousands of years. And one of them is a direct hybrid. If contact between Neanderthals and Denisovans had been vanishingly rare, we should not have drawn that card so early. This is where a careful viewer should hold the conclusion loosely, because there are 2 honest readings and the field has not closed the gap between them. One reading says the odds speak for themselves. Encounters must have been ordinary, and the Altai was a place where 2 kinds of human overlapped for a long time. The other reading says that Denisova Cave is one specific site, sitting at the geographic seam between 2 ranges, sampled far more intensively than anywhere else on Earth. and that finding an unusual thing at the exact spot where the search has been most intense is not the same as that thing being common. We have one first-generation individual, one is a fact, one is not a frequency. What Denny does establish, beyond argument, is that these were not sealed categories. Two populations separated for hundreds of thousands of years met in a valley in Siberia and had a child who lived to adolescence. Hold one number against everything that follows. At this point in the story, a decade of headlines. A species in every textbook, the entire physical record of the Denisovans would fit in a coat pocket. A fingertip, a couple of teeth, a splinter of arm bone. That is the whole body of evidence for a people who ranged across a continent. Everything so far comes from a single cave, which raises the obvious question, and the answer, when it finally came, moved the Denisovans across a continent and up onto the roof of the world. Number 2. The jaw on the plateau and the jaw under the sea. Baixia Karst Cave sits in Jiahe County, in Gansu Province in northwestern China, at more than 3,200 meters above sea level. It is a Buddhist sanctuary. Around 1980, a monk in the cave found a piece of a lower jaw with two large molars still seated in it, and it was eventually passed to researchers at Lanzhou University. It sat more or less unexamined for decades. In 2019, Fa Hu Chen, Dongju Zhang, Jean-Jacques Hublin, and colleagues published it in Nature. There was no usable DNA in it. What they did instead was drill into the teeth and read the collagen, the proteins themselves, which survive when genetic material does not. The amino acid sequence carried variants that matched Denisovan and did not match Neanderthal or modern human. Uranium series dating on the carbonate crust gave a minimum age of about 160,000 years. So, a Denisovan on the Tibetan Plateau at altitude 160,000 years ago. Modern humans do not appear on that plateau in any numbers until tens of thousands of years later, and when they do, they struggle with it. The plateau result did not stay alone. In 2020, Dongju Zhang and colleagues published Denisovan mitochondrial DNA recovered from the cave's sediments in the journal Science, DNA from the dirt itself, no body required, and in 2024, the same cave produced a rib, published in Nature, from a much later layer, showing the site was held by these people across an enormous stretch of time. That study was not really about the rib. It was about what lay around it. Layers dense with butchered animal bone. The ordinary residue of a group that hunted the plateau, processed what it killed, and came back to the same shelter to do it again. Not a refuge visited once in a crisis, a home address. Then the map broke open again. In a direction nobody had drawn in 2010, a fishing trawler working the Penghu Channel off the west coast of Taiwan dredged up a heavy, thick-set lower jaw from the seabed. During the ice ages, that channel was dry land. The fossil went into a Taiwanese museum as an oddity. Clearly archaic, unclearly anything. In April of 2025, Takumi Tsutaya, Enrico Cappellini, and an international team published their analysis in Science. Using the same protein reading approach, they recovered 4,241 amino acid residues from the jaw, including two variants specific to Denisovans and one that identified the individual as male. Siberia, the Tibetan Plateau, subtropical Taiwan. 3 climates, 1 people. The honest footnote on the Taiwanese jaw is its age, and the paper does not hide it because it came off the seabed rather than out of a dated layer. The dating rests on sea level history and associated fauna, and the result is not one number but 2 possible windows, very roughly 10 to 70,000 years ago, or 130 to 190,000 years ago. That is not a rounding error. It is a genuine open question, and it matters, because one of those windows puts Denisovans in the tropics at the same time modern humans were moving through. We now had jaws, teeth, a rib, a fingertip, sediment, and a range spanning half a continent. We still did not have a face that changed in a way nobody planned for, because of a decision a laborer made in 1933. Number 1, the skull in the well and the gene in the blood. In 1933, workers were building a bridge over the Songhua River at Harbin in northeastern China. The region was under Japanese occupation. One of the Chinese laborers on the bridge pulled a human skull out of the ground, a very large, very complete cranium, heavy-browed, with a single molar still in place. He did not hand it to his employers. He took it home and dropped it down an abandoned well, which is an old and practical way of hiding something valuable in that part of the world, and he told no one. He kept that secret for the rest of his life, 85 years. Shortly before he died, he told his grandson, In 2018, the family retrieved the skull and gave it to Chiang Ji, a professor at Hebei Geo University. In 2021, Ji Sijun-ni and colleagues published it in the journal The Innovation. It was a magnificent specimen, one of the most complete archaic human crania ever recovered, and they gave it a new species name, Homo longi, and a nickname that traveled faster than the paper did, Dragon Man. At the time, several researchers looked at that skull and said, more or less openly, that it looked like the Denisovans skull we did not have. But looking like is not evidence. The specimen had been out of the ground and in a well for the better part of a century, and repeated attempts to pull DNA out of its dense bone and its tooth came back with nothing usable. In June of 2025, a team led by Qiumei Fu at the Institute of Vertebrate Paleontology and Paleoanthropology in Beijing tried the last thing left, not the bone. The plaque dental calculus is hardened tartar, the mineralized film that builds up on a tooth in life and then locks whatever it trapped into place. They scraped 0.3 milligrams of it from the blackened root of that single remaining molar. From that, they recovered mitochondrial DNA. It matched the Denisovan sequences from Siberia. A companion study published in Science the same month read the skull's proteins and reached the same conclusion from the other direction. The skull is at least 146,000 years old, and after 15 years of a species defined by its absence, we could look at one. Now the caution. And it is not small, because this is exactly the kind of result that gets flattened in the retelling. Nearly 4/5 of the human genetic material recovered from that plaque sample was modern contamination, handling, storage, a century of human contact. The Denisovan signal is the minority fraction, isolated by the damage patterns that distinguish genuinely ancient DNA from recent. That is a well-established method, and the authors applied it properly. It is also a thin thread to hang a face on, and it deserves saying plainly. The naming is not settled either. Fu's team places the Denisovans within Homo longi. In 2024, Christopher Bay and Xuje Wu argued in Nature Communications for a different arrangement entirely, proposing the name Homo juluensis for a group of large-skulled Eastern Asian fossils and putting much of the Denisovan material there instead. Two teams, the same bones, two different names. That argument is live right now, and then there is the part that is not an argument at all. In 2014, Emilia Huerta Sanchez and colleagues published a study in Nature that had started as a straightforward question about high-altitude physiology. The Tibetan Plateau is a hostile place to live. Above 3,000 meters, the standard human response to thin air is to manufacture more red blood cells, which thickens the blood, which over years causes strokes, heart failure, and pregnancies that end badly. Tibetans do not do this. They carry more oxygen without thickening their blood, and they have done so for a very long time. The team resequenced the region around a gene called EPAS1 in 40 Tibetan and 40 Han Chinese individuals. EPAS1 governs the body's response to low oxygen. What they found in the Tibetan samples was a stretch of DNA about 33,000 base pairs long with a haplotype structure that could not be explained by ordinary mutation and selection. It looked like something that had arrived from outside, already assembled. They compared it against the Denisovan genome from the cave. It matched. Follow-up work by Xinjun Zhang and colleagues in the Proceedings of the National Academy of Sciences in 2021 traced its spread. That Denisovan-derived haplotype is carried by around 72% of Tibetans and by about 2% of Han Chinese, and its frequency across Himalayan populations rises with the altitude they live at. Sit with the sequence of events. A population we cannot name with confidence, whose skull we only identified last year, was living on that plateau at least 160,000 years ago. They adapted to it. Modern humans arrived far later, met them, had children with them, and inherited the solution ready-made. The answer to a problem our species had never had time to solve. If you have ancestry from the Himalayas, this is not history. It is running in you right now every time you take a breath at altitude. And it is not only there. Papuans carry roughly 5% Denisovan ancestry, and a body of work on those genomes points to immune system genes among the inherited material. Across Asia and Oceania, hundreds of millions of people carry pieces of them. We spent a century looking for the missing Denisovans in the ground. They did not go missing, they were absorbed. Here's what the whole sequence adds up to, and it is not really a story about a lost species. Consider how we met everyone else. Neanderthals, skull first in 1856, then a name, then a century and a half of argument, then finally a genome, Homo erectus, Homo floresiensis, the same order, bones, then a name, then biology, if we were lucky. The Denisovans ran the film backwards: genome, then population, then a hybrid child, then a jaw, then a range across a continent, then 92 years after a man dropped a skull down a well to keep it away from an occupying army, a face. Nothing in the ground changed. Every one of these objects was already sitting in a drawer or a cave or a museum basement before we could read it. The Harbin skull spent 85 years in a well and then 7 more years in a laboratory, being called a probably. The XIA jaw sat in a collection for nearly 40 years. The Taiwanese jaw came up in a fishing net and waited 15. Denny was in a tray of unidentifiable splinters. What changed was not the evidence. It was our ability to read it. Collagen, sediment DNA, single-stranded libraries, 0.3 of a milligram of tooth plaque, which raises the uncomfortable version of the same thought. If the Denisovans were invisible to us for a century while sitting in our own collections, the honest position is that we do not know how many other populations are currently filed as unidentifiable, waiting for a method that has not been invented yet. The gaps in the human family tree may not be gaps in the record. They may be gaps in the reading. And the last piece is the one we have to hold most loosely. We still do not know whether Denisovan means one people or several. The Papuan genomes say at least 2 deeply separate lineages. The bones say Siberia and Tibet and Taiwan, which is a range no single population has any business covering. It is entirely possible that we have given one name to a set of peoples who never thought of themselves as related at all, and that the tidiest thing about the Denisovans is the word we invented to stop having to think about how little we know. You are, in a measurable sense, part of the evidence. That is a strange inheritance to be handed. It is also the only kind any of us actually get. If you want to see more of what the record keeps hidden, click the video on screen now and make sure to subscribe.